Can you see my screen?
Very well.
Hey guys, you have to unmute. Keep it quiet. We're just 8 of us here.
Yes, hata wa unmute. Gauss ni nani?
Ati?
Ni nani we madouble M mob?
Sijue.
Ni internet yenu
I think ni internet yako.
Tuanze basi. So let me retrieve the specific topic. Let me see. We see out of the scope of what was supposed to be covered. First one is just basic introduction, like to bacteria. Just a second. Yeah, bacterial classification, structure, and function. Yeah, so I hope Nini. Okay, now let us start. Bacterial classification, structure, and function. So classification of bacteria, I believe at least at the very least we remember, right? Gram positive and gram negative, and this classification is based on the cell wall. Then on that note, kuna the others, we call them atypical organisms. Then bacterial structure, now upon you to spend maybe some 20 minutes before we go to the function, growth, and metabolism. Yeah. So tuanze na nini? Tuanze na how the bacterial cell looks like. And this is a good image. You can see, number one, there is no nucleus. You know, in the eukaryotic cell, we see the nucleus being on its membrane, right? But for bacteria, we are seeing there is no nucleus. What we have is just this nucleus. And when we are looking at bacterial transcription, tulisema that DNA to RNA, that transcription from DNA to RNA, it happens in the nucleoid. The nucleoid is a place where the DNA, or rather the chromosomes, are concentrated. But if the nucleoid is not there, then the answer would be in the cytoplasm, because the bacteria has no defined nucleus, okay? So you're seeing the nucleoid region, then we have the cytoplasm. In the cytoplasm is where we'll have the cell organelles. Ribosomes, we are seeing the ribosomes there. Then there is a plasma membrane, same as humans, it has the plasma membranes. Then what is unique about bacterial cells is that they have a cell wall. They have a cell wall. And then the other things, flagella, we are seeing flagella here, the extensions that they use for movement. We're seeing the pili, we'll see them when we're talking about even things such as conjugation, or rather, in conjugation, we'll see how can bacteria communicate with each other, such things. So just for the structure of the bacterial cell is, just know that as compared to the eukaryotic cell, we don't have the nucleus being on its own. Okay. Then the cell wall. Looking at the cell wall specifically, we'll also look at this when we're talking about mechanism of action of penicillins and such things. But because we said we will combine microbiology and pharmacology and clinical pharmacy, which is impossible to do in 1 week. I'll be mentioning just the highlights. So for the cell wall, we call it, it's made up of peptidoglycan. It's called the peptidoglycan layer, and it's made up of two alternative sugars, that is N-acetylglucosamine and N-acetylmuramic acid. N-acetylglucosamine and N-acetylmuramic acid. You can see here they have been linked. N-acetylmuramic, N-acetylglucosamine in that way. And then there is this a few amino acid. Then again, I think this is a typical structure here. And in short, this whole structure, we call it the peptidoglycan layer. The peptido part comes from this peptide chain. These are amino acids. Amino acids here. You can see alanine, some amino acids here. Hence the name peptide. Peptidoglycan is now the sugars, right? These are sugar structure from the biochemistry we did last week. So peptidoglycan layer. And just atuja cover, but we know that in gram-positive, it has more peptido, or rather it has a thicker peptidoglycan layer as compared to gram-negative. And now in the peptidoglycan layer, that is where drugs such as penicillins, cephalosporins, vancomycins, that's where they work. They inhibit this formation of the cell wall. So once they inhibit this formation of the cell wall, we are seeing, once we've inhibited formation of this wall, we're seeing this wall acting like a barrier. So if we have inhibited formation of this cell wall, it means that cell is more accessible, number one, to our immunity. Remember, when we're looking at antibiotics, we say that one of the properties of an ideal antibiotic is that it works hand in hand with our immune system to clear the infection. So imagine if we remove this cell wall, the bacteria is now well exposed to be phagocytosed by the white blood cells and such things. The cell membrane, same as humans, it's just that then this is the structure we were talking about. Gram-positive, you're seeing the amount of the peptidoglycan layer is thick there, right? Whereas in gram-negative, we are seeing a very thin peptidoglycan layer. So that is why drugs that act on the cell wall, will see them mostly being used for treatment of gram-positive infections, right? Things like penicillin, we'll see them treating staphylococci, streptococci, right? Yeah. So in this structure displayed here in the first page, this is a typical structure of a bacterial cell. However, it's more typical for a gram-positive organism. That the first thing we have is the cell wall, then the plasma membrane, then the cytoplasm. Because when we come to gram-negative organisms, we are seeing, number one, they have an outer membrane. Then we have the peptidoglycan layer. Then we have the periplasmic space, after which we have the cytoplasmic membrane. Now, a question that is asked is, why are gram-negative a bit more resistant to infections than gram-positive? It's because the gram-negative organisms, they secrete enzymes into this periplasmic space. These enzymes break down the antibiotic before it even reaches the cell itself, right? So, this periplasmic space really plays a huge role. Then in gram-negative organism, what we can see is that, remember we said it's outer membrane, peptidoglycan, a periplasmic space, then the cytoplasmic membrane. So the outer membrane, it contains the endotoxin, the lipid A, or rather together here, it's called combination of the lipid A and the O polysaccharide. The O polysaccharide is antigenic, then the lipid A is an endotoxin. Endotoxin, in terms of lipid A, leads to your immunity acting on it, raising your body temperature. Then we call them pyrogens. Yeah, pyrogen is the right term even. Lipid A is a pyrogen, right? The O polysaccharide is the antigenic portion, but lipid A is the actual pyrogen. Anything that raises your body temperature. So here is the point I mentioned, that the periplasmic space, it contains enzyme and other substances. Then here we already say the peptidoglycan layer in gram-negative is thin, and the outer membrane is distinguished by the presence of the lipopolysaccharide, LPS. Now the lipopolysaccharide, that tells us lipid plus a polysaccharide. The lipid is lipid A, then the polysaccharide is this O polysaccharide. So when you ask which one has a lipopolysaccharide, bring them positive and negative, that is negative. Then we can also look at the flagella. The flagella, we know it's important for movement, right? Here it is. Flagella is just think of the flagella like a wagging tail, but once the bacteria is wagging its tail, it will move forward, right? So just think of the flagella like a wagging tail. So cells can have one flagella or many flagella. We'll see when we are looking, especially at gram-negative, we'll see you can distinguish them based on the flagella. Even the arrangement of the flagella. Yeah. The other thing we mentioned is the pili or the pillars. They are shorter, they are thinner than flagella. While flagella is for movement, the pillar number one is for attachment, right? Attachment, and they also promote cell-to-cell contact. When we said we will see how the bacteria is able to communicate, how can one bacterial cell transfer its resistance to another bacterial cell? We said in biotechnology, when they exchange plasmids, right? The one with resistance gives the one without resistance a plasmid, and we all know what plasmids are. So again, we go there. So let's go to spores and sporulation, because for the spore, I don't know if anyone can remember any one organism that actually produces spore, or even if they can remember what a spore is.
Is Clostridium spore? I don't know.
Yeah, Clostridium. So essentially, what spores are, it's a survival mechanism. Think of spores as the tortoise, the shell of that tortoise. The way once the head of the tortoise goes inside the shell, it's like it's protected. It's a survival mechanism for the bacteria, but very specific bacteria, especially the Clostridium family is one, and the other one is the Bacillus cereus that causes gastroenteritis, things that form spores. So why would bacteria form a spore? Now, for a spore, it's like think of the bacteria. It's somewhere there is no food, there is no water, there's bacteria. So it has two fates. Number one, it's either it dies because it has no food, water, or nutrients, or number two, it adapts. How does it adapt? It forms a spore, something that a version of it that does not, or rather you can say does not need energy to survive. And then uniquely, when that spore senses water, senses presence of nutrients, the spore comes back and comes back to life and produces the bacteria, right? So it's very unique. Let's look at a typical example of Clostridium tetanus, right? We always mention tetanus. That, when someone is hurt, someone was tilling their garden, something of the sort, and then they cut themselves. We always say that person should go for TT injection, tetanus injection, right? But why? Why? It's because number one, the tetanus spores were in the soil, right? They were harmless. When they were in the soil, they were harmless. They couldn't do anything to anyone. They had no source of nutrients per se, right? But when this person cut themselves and the spores entered into the system, the spores detected number one, blood, water. Hey, they said, yo, ladies, we are home. They said, my guys, I can see water, I can sense this and this. And so the Clostridium tetani comes back in full force. And after that, we all know how tetanus can kill you, right? Yeah. So spores, essentially, it's when the environmental conditions are harsh, too harsh for the survival of the organism. Hence, that organism forms a spore. And we've only mentioned number one, the Clostridium family, anthrax, that is Bacillus anthracis, and Bacillus cereus. It's just these few organisms that can actually do this. So there's the process of forming a spore. I'll just work with this diagram, but try to highlight the main points. So, or just another thing that maybe I had mentioned is that spores, number one, they are very resistant to heat. They survive boiling, desiccation, UV light, and even chemical agents, right? That it's only sterilization procedures are assessed by the ability to kill spores. So if you want to know this sterilization procedure is legit, we'll bring a spore of Clostridium, a spore of Bacillus, and see if it can actually kill it. Yeah. So sporulation, that is the process of forming a spore, essentially is just simply put, it's repackaging the DNA. Remember, the DNA is it's the basic code of a living being. DNA is the basic code of a living being. When like today when we were learning sperm and ovum, when a sperm and ovum fertilizes, the zygote starts as one cell. What the zygote just has is its DNA. It starts as one cell just with its DNA. But from that zygote, a whole human being is formed, telling us that for us to, and that is the principle that even scientists are using, for us to clone a person, for us to clone a whole human being, a whole animal, essentially, all we need is the DNA, the basic code for a living being, right? That's all we need. And it's the same with sporulation. Sporulation, you can say bacteria, they are very smart. They already figured this out. They know that so long as we have our DNA somewhere, we can always grow from the DNA. So sporulation is just, you take a copy of the DNA, you package it into a form that literally needs negligible amounts of water and energy, right? Very, in short, something that needs the basic minimum to survive. So spore formation is just here. Now we can work with the diagram because we've said number one, it's just a matter of having a copy of your DNA somewhere, right? So here we are. We said the nucleus is where the chromosomes are concentrated, right? Now you can see there's some invagination, or rather some separation. Let's just say separation, right? We separate the more of the cell into two parts. Now, and we make sure this second part. Now, I'll be focusing our efforts on this second part, because this second part is where we will form our spore from. This second part, we make sure it has a copy of the cell's DNA, right? So once it has the copy of its own DNA, we will separate it. We'll just separate it, and what makes it more resistant to this thing that has heating and UV radiation is now highlighted here. That that spore coat, what is it? Yeah, so the retains the complete machinery for protein synthesis and new synthesis. Okay, so it has a tough spore coat. So in short, let's think of it as a jacket that we ensure this thing has a heavy jacket. Remember, if for example, if let's say Devayani is in a condition whereby there is no nutrients, no water. She knows her fate is death, but she knows she can separate her DNA of water. She can be able to grow from that DNA. She will package her DNA somewhere in a box and make sure that box is really sealed, right? She'll put jacket or lead, whatever amount of coating to make sure her DNA is safe. And it's exactly what bacteria does. We call it a spore coat, whereby it will be engulfed in a tough coat. And now this tough coat is what will make it resistant to heat, UV radiation, and even chemicals, all those things, right? And now here is when we say that once that spore, now that spore has senses that there's enough the nutritional requirements, there's water, there's food, right? Once it senses that, the spore germinates, right? Whereby it is water and it's able, the bacteria is able to grow again, right? So just imagine sporulation like a tactic of preserving oneself. Putting your DNA somewhere in a box and making sure that box can never be broken into. Then the day that you know that it's safe to come out, you come out and you're able to, number one, replicate your DNA. You're able to use your ribosomes to synthesize the proteins that you need for survival. And here it is that I don't left out, that the spore retains the complete machinery for protein synthesis, right? Meaning, remember, central dogma of molecular biology: information flows from DNA to RNA to proteins. So if we preserve our DNA, but we have no ribosomes to make the proteins we need, we will just have some DNA somewhere. So the bacteria retains the capabilities to synthesize the proteins. So just ensure you know this. You need the Clostridium family, the Tetany, Perfringens, Botulinum, and Bacillus. That is the anthrax, Bacillus anthracis, and Bacillus cereus, which causes food poisoning. Bacillus cereus, from this name even, cereus, that tells you cereals, right? So when you're in rice is where you find these spores. Yeah, they are found in rice. So you eat bad rice, you find they had these spores, they reach your stomach, the environment is favorable, the spores, they get out in full force, and then we say you have food poisoning, right? So Bacillus cereus is here, and all of these are gram-positive organisms. Specifically, they are gram-positive rods. Remember, we look at cocci. Cocci is essentially spherical shape. Rods, we say it's more of a rectangular shape. Growth and metabolism. Here, I just want you to take, this curve. This curve is everything. Yeah. Or it's called a curve, or this graph. I think graph is the right term. So this graph is everything. We have the lag phase, the log phase, the stationary phase, and the death phase. This is the growth of a bacteria. So in this case, I just want you to imagine you have Staphylococcus aureus on your skin, right? We know that it's part of the normal flora. So you have Staphylococcus aureus on your skin. We take that Staph aureus, we make our media, and we put a few cells of Staph aureus in that media. The first thing Staph aureus will do, or even the first thing, even you as a person, for example, in first year, let's even work in first year. In first year, you guys didn't know each other, right? Yeah, when you came in your first, second classes, the first thing was to read the room, see what people are saying, such things, right? So you don't just enter a room full of strangers and you're already shouting blah blah blah. No, first of all, you enter and read the room, and that is what bacteria does. And this is what the lag phase is. The bacteria is adapting to its environment. It's like, oh, this is USIU. Higher people buy coffee at Java, and you are from UN, like Stanley. They don't even drink. They don't even have coffee in UN. See? So first you read the room, see how it is. Then now the second phase, which is the log phase, after the bacteria has adapted to the environment, now the bacteria can grow. Right? It can grow. But remember, our picture is still in this media. Remember, we're having media in Petri dishes. Now our bacteria is growing here. Our bacteria is thriving in the log phase. It has adapted to the environment. Now it is growing. Now it reaches a place whereby the bacteria has grown to the point of let's say it has, due to the, remember, since this is a media, right? We have limited resources, right? We have limited resources. And that's why I want you to think of it in terms of a media, not in terms of it's in your body, because in your body for a bacteria, there's no limited resources. Yeah. So it reaches a point whereby there's this stationary phase. This stationary phase is, you can say it's the point of, I don't know, is it the word balance? I don't know the right term to use for this. I don't want to say balance accounts. Point where not growth and death are equal. Let's say it's at its optimum. I don't know. Optimum in terms of that the nutrients that are there, it's just enough for the cells that are there. In short, there's no more growth here. There's no more growth. That now the bacteria, it's like they are enough in the room, right? They're enough in the room. But as time goes by, there's accumulation of toxins. Remember, these bacteria are metabolizing kina glucose, kina amino acids. And we said, as much as this process releases energy, we also said it releases toxins, things such as ammonia, right? And now in the death phase, it's that toxins, number one, they have started to build up in that media. Number two, now the bacteria are actively competing for limited nutrients because they are finished. The nutrients are finished. So they are competing for nutrients. So one is not able to get nutrients, it starts dying and all that. So know the four phases. The lag phase, you can just say here the cell is adapting to the environment. The log phase, the cell is growing. The stationary phase, where the cell is the cells is the right term. The cells are no longer growing, they're in a constant state. Then it reaches a point, the resources are limited, toxic waste starts to build up, and now cells starts dying in this phase. So lag, log, don't confuse between the two. Lag A, then log O. A comes before O. So lag, log, stationary, then death. The log phase is also called the exponential phase. Okay. Then for energy production, let me see if it's bacterial growth and metabolism. Okay. You can say it's under metabolism. So for energy production, good thing is that we've looked at how energy is produced by humans. So here it becomes quite a bit simpler. So, and again, we'll just go to the diagram. Yeah. So you can see bacteria just uses the same mechanisms as the ones we covered. Fermentation, whereby in this case, it's an anaerobic process. So once you give it glucose, glycolysis as usual, but the end product is right. Then in aerobic respiration, still produces the NADH from glycolysis from TCA cycle. We are seeing TCA cycle here. Again, we can see the electron transport chain here coming in. Production of water. Remember, we said the electron acceptor in the electron transport chain is oxygen. Yeah, and we're able to produce water. So that is energy. In energy, so here in energy, in terms of energy, know that bacteria can undergo three types of three mechanisms for energy production. That is aerobic respiration, whereby presence of oxygen. Then we also have anaerobic respiration, right? Anaerobic, I think we all know what anaerobic means, that we don't have oxygen. Then in anaerobic respiration, there's two types of organisms. Number one, there is the ones we call facultative anaerobes. Facultative anaerobes, what it simply means is that if there is no oxygen, this thing can undergoes anaerobic metabolism effortless, right? But obligatory anaerobes. Obligate means that this organism only undergoes anaerobic metabolism. Okay, it only undergoes anaerobic metabolism. So, and you'll also say, in this case, you will, once we are starting to look at mechanism of action of metronidazole, things such as those, we'll see how obligate anaerobes are easily killed, right? Because for obligate anaerobes, think of it in terms of, it's like oxygen is toxic to this stuff, right? For a facultative anaerobe, oxygen can be there, cannot be there. Facultative anaerobe is like, yo, me, I can do with it either way. But for an obligate anaerobe, that one does not want oxygen. Obligate anaerobe, if you introduce oxygen into the mix, it gets out of the mix. Then another, just a minor one, is fermentation, which is an anaerobic process, just exhibited by specific bacteria. And fermentation, where we utilize fermentation when even we're making alcohol, right? But it's just simply that conversion of glucose to alcohol, as I've said. But the two major, it's aerobic and anaerobic. Cell wall synthesis will look at this more when we are looking at penicillins. But I hope you glucosamine, yeah, that the peptido part comes from these amino acids linking these layers. And one of the enzymes that plays a crucial role in formation of that peptidoglycan layer is which enzyme? Transpeptidase enzyme, right? And that is the enzyme that penicillins come to inhibit. That transpeptidase is important in interlinking here in the peptidoglycan layer. But we'll see more of that when we're looking at penicillins. We already say beta-lactam targeting this part, even vancomycin, bacitracin. Yeah. So I think looking at these questions, there's a bacterial culture with a starting density of 10 to power 3 is incubated in liquid nutrient broth. If the bacteria have both a lag time and a generation time of 10 minutes, what will be the cell density at 30 minutes? That the lag time, right? The lag time, the bacteria is not growing, right? It's adapting. It's just looking around, right? Taking you back to the graph here. Here, in the lag phase, the bacteria is not growing, but in the log phase, it grows. So if you come back to the question, even without us actually calculating it, if you come back to the question, we've been told that the bacteria has a lag time of 10 minutes and a generation time of 10 minutes. Then we've been asked what will happen at 30 minutes or after 30 minutes. So we'll say the first 10 minutes, the number of bacteria will still be 10 to power 3, right? It will still be 10 to power 3. Then after 10 minutes, now we will enter our replication phase, our log phase. And that is where the bacteria, number one, it will double for the first 10 minutes. Then that that is our minute 20. The bacteria has doubled, right? Then after another 10 minutes, the bacteria will have double again. So that is 2 times 2, right? Because it's doubling. So 2 times 2, that is 4. So it will be 4 by 10 to the power of 3. And so the first 10 minutes, it's in the lag phase. It's not dividing. Then log phase, we've been told 10 minutes. Then in this question, which components are found in the cell wall of gram-positive but not gram-negative? So which is found in the cell wall of gram-positive but not a gram-negative organism? We skipped this part, but because this is a common question, let's just go back to this: steatoic acid, lipoteitoic acid. But at least I know for lipopolysaccharide, and all of you can say this one is definitely in gram negative, right? That one we've covered it. Lipopolysaccharide is in gram negative. Then we've also said peptidoglycan is in both gram positive and gram negative, right? It's just that in gram positive it's a lot, in gram negative it's not so much. Okay. Yeah, cytoplasmic membrane. For cytoplasmic membrane, we'll say what about the cytoplasmic membrane? That they all have that cytoplasmic membrane or plasma membrane. Remember, this, the cytoplasmic membrane is just the plasma membrane. Then the outer membrane, right? The outer membrane, it's only also again gram negative that has an outer membrane. Remember, gram positive is just peptidoglycan, cytoplasm, but gram negative is outer membrane, peptidoglycan, periplasmic space, then gram, then the cytoplasmic membrane or the plasma membrane. So just
just a minute, guys. I have something to add.
See that part here.
You can go
Yeah.
back to those questions.
To the question. Okay.
Yeah. So in case if there is a question which says porine on the outer membrane, it is only positive on the gram. It's only there in gram negative. Gram positive doesn't have a porine on outer membranes.
Yeah.
In case if there's such a question that comes up.
Thank you, Shina. Thank you. So for gram positive, just because I left that out, just to highlight that the teichoic acid, which is essentially a polymer of just that. Now, just on this, because they like bringing this question to confuse someone. There's teichoic acid, right? And if anyone can remember Obila's voice right now, lipoteichoic acid, right? Teichoic acid and lipoteichoic acid. Now you can be asked, what's the difference between lipoteichoic acid and teichoic acid? I feel like I'm saying the same thing. Okay, let me say it again to make sure I've said it. What's the difference between teichoic acid and lipoteichoic acid? So lipoteichoic acid, let me see if let me project the screen, the diagram, so that it's easy. It's just that lipoteichoic acid, from the word lipo, it tells us there's a lipid somewhere, right? While teichoic acid, there is no, there is no lipid, right? So don't know, my internet is misbehaving, but I'll just say teichoic acid, that acid will be anchored onto the peptidoglycan layer. But for the lipoteichoic acid, the acid is anchored onto the cell membrane where we are having our lipids. Let me project this for clarity. So I hope so far we are getting we're getting. So here it is: teichoic acid and lipoteichoic acid. You're seeing this teichoic acid, it's anchored on the, here it is, it's anchored on the peptidoglycan layer. here is a peptidoglycan layer. It's anchored to the cytoplasmic phospholipid bilayer. So teichoic acid and lipoteichoic acid. But you can see they are all found where in gram. We just have the lipopolysaccharide as displayed here, right? We just have the gram. We just have the lipopolysaccharide. But gram positive, we have right now. At least if we say that we list, even if it's just 3 differences between gram positive and gram negative, we can say gram positive have more peptidoglycan, negative less. Number 2, gram positive has the teichoic and lipoteichoic acids attached onto it, but gram negative has the lipopolysaccharide, right? Again, we can say gram negative has the outer membrane, whereas the gram positive has no outer membrane, right? At least we can list those 3 before that we've covered. Then here, this one is more of a bit clinical application, whereby we're talking about this Clostridium botulinum. Remember, the process of sporulation is just the essential thing to remember with sporulation is that we are taking our DNA and putting it somewhere safe, that once the conditions are favorable, we will be able to rise again, right? So looking at bacterial genetics. Genetics will just take us 10 minutes. Then we can take a break. We look at identification. Most for identification, we'll just be doing gram staining. Then we can dive right onto gram positive organisms. Yeah. So we look at the genetics. Genetics, good thing is that we've covered a bit of it while looking at biotechnology, right? If anyone can remember biotechnology, we said we mentioned these things, of plasmids, right? In your own words, you don't have to define it as it is. Like, what was the difference between the plasmids and the, let me call it, the conventional DNA of an organism? Okay, so in short, for plasmids, plasmids were just, we say they are extra chromosomal DNA, the chromosome, right? Yeah, it was just extra chromosomal DNA. I think that's the simplest way to remember plasmids. And that's why you can see plasmid is always here by itself. It is your plasmid, but you have your chromosomal DNA here in the nucleoid region. Please remember again, nucleoid region. In bacteria, we don't have a defined nucleus like the one we have in eukaryotic cells. We saw eukaryotic cells when we were studying biochemistry, right? In our cells, number 1, we have the cytoplasmic membrane around the cell. We have the nuclear membrane around our nucleus. We even have a mitochondrial membrane around our mitochondria, and all these are phospholipid bilayers. But with bacteria, bacteria just said, I'm just doing the one. And the one is just the cytoplasmic membrane. But you can say for gram negative decided to do 2, cytoplasmic membrane and outer membrane. Remember, gram negative has an outer membrane. So looking at the just bacterial genetics, we've already said we know what a chromosome is, right? And we said the plasmid here, it is an extra chromosomal DNA. So applications in biotechnology, use insulin from E. coli, right? Yeah, so there's we won't look at bacteriophage as much, but I'll just mention something about bacteriophage. Number one, bacteriophage, you know that name is sometimes is term line starts with the deceived. Yeah. So bacteriophage, it's actually a virus, right? It's a virus that can infect what? Bacteria, right? Once we cover viruses, we will see viruses they come, they enter our cells. Viruses, we say they are now inside your cell. If COVID-19 does not enter inside your respiratory cell, COVID-19 dies, right? So viruses, they're cellular organisms. Same, so bacteriophage, it's a virus. At its core, it's just a virus. But it's a virus that infects bacteria. Beginning is even when it's like we're revising a bit of virology. The virus touches, and then it enters. Once it enters, it's a nucleic material, whether it's RNA or DNA, enters. It's taken up by the host cell. Now, in this case, the host cell is the bacteria, and the host cell, the virus protein, the phage proteins, and these phage proteins, they eventually, I guess, I really skimmed through biotechnology, so when I get the chance, I throw you there once again. Bacteriophage can be used virus, right? This bacteriophage, here it is, it has left its DNA here. That DNA has come to the bacteria and translated to form protein. So that tells us if we take a bacteriophage, right? If we take a bacteriophage and we give it in its DNA, we encode the code of making insulin. We can make the bacteriophage infect the protein, which is insulin in this case. So bacteriophage can be used as vectors. Yeah. Then the other thing we also mentioned last week was cosmid. This is whereby you take 2, 1. So that was the definition of a cosmid. Just take technology on your own. Finally, we will finish with gene transfer. Gene transfer. Why is gene transfer important? Especially right now, when the era of MR is the next pandemic. And you wonder if it's the next pandemic, why can't preparing ourselves for this pandemic? Professional note for all of you interested in AMR. I don't know which European association. Kenya has recently been given a lot of money to combat the AMR. So a piece of the money. Yeah. So gene transfer from the tab. Gene gene, we know, something that is for a protein, right? Then transfer is literally giving someone between. So genes can be transferred from one bacteria to another. And here you just see me repeating, especially in terms of antimicrobial resistance, or rather how, for example, you have one E. coli cell that is not resistant to, for example, nitrofurantoid. Then we have a neighboring E. coli cell that is resistant to nitrofurantoid. And then we leave them to chat kiasi. They start laughing, and before you know it, the other cell that was not resistant has become resistant. That tells you something happened between these two. They transferred something between them. What did they transfer? Most likely it's what? Information, right? For example, the information in this case would be what? The resistant one tells the sensitive one, yo, you know me, nitrofurantoin does not kill me. The sensitive one is like, oh, really? Like, yeah. How to enter the cell, and then I break down that thing before it can harm me. So that's how you that's how you make sure it doesn't kill you. Yeah. Okay, that's information. Yeah, and then I take you back to central dogma of molecular biology. Information flows from DNA to RNA to proteins. So the DNA information is originally in the DNA. So we need to transfer these genes. And there are three major mechanisms. Number one, we have conjugation. Number two, transduction. And number three, transformation. Conjugation, it's those type of questions that they are tested. So we look, the good thing with this, Nene, is that it has these diagrams that can help here. Transduction, I don't know if it does, and then for transformation, we'll definitely see. Okay. So let's start with conjugation. We've already said we are transferring one gene to from one cell to another cell. Now, because in nature we always have to you can say denote or rather assign identity. We say the donor is male and the recipient is a female cell. I don't know, they can decide to ask which one is the donor. You know, these people. The donor is a male cell and the recipient is the female cell in conjugation, right? So in conjugation, essentially, there's a sex pili that is used to make contact between the two cells. And we say they are very important for attachment. And we also say the other thing, they're important for communication. And that is what we're coming down here to, communication. Now here, because we call it the sex pili, that's why we're saying male and female. I do not wish to attack anyone in the audience. This is microbiology. So this anime, where let's just go to the diagram. It's easier to see with the diagram. So let's look at these two cells. Now the F plus and an F minus. The F plus is the one that has the F plasmid. Okay. The F minus does not have a F plasmid. So you can see number one, they've connected, you know, like, bro, what's up? What's popping? Then once they connect, you can see now it has become like one cell, right? There's no cell membrane separating these two things, right? There's no cell membrane separating these two things. And remember from Nini, we said plasmids, they're just extra chromosomal DNA, but they're also what here? Double-stranded. Same like the normal DNA, double-stranded. Even the plasmid is double-stranded. Don't hate on the plasmid. The plasmid is not a little guy, it is double-stranded. So here, and that double strand, it's a huge deal. Here it is. One of the strand gets nicked and decides, me, I'm going this way. It goes that way. And then once it goes that way, there it is. It has entered now the F minus cell. And from this strand, the bacterial cell is able to synthesize a complementary strand so that it is double-stranded. And then at the end, you see that they've separated, right? So in short, these cells, they came into contact. Plasmid, one strand of the plasmid went to the other side. They both synthesized a complementary strand, and then they were like, bro, I've shown you how to fight nitrofurantoin. Are you good? He's like, I'm good. Let that nitrofurantoin comes. What a bit of the terms we use, because that's the story. I wanted you first to get the story that it's two cells, they come into contact. The one with the plasmid to be exchanged gives it one strand. They both synthesize a complementary strand, and that is it. So all of this, what makes the mixed contact is here. The presence, the process requires what? The presence of the donor cell of what? Of the hair-like called sex pili. These are the ones that make contact with creation of DNA transfer. So in short, remember we said we have the male and we have the female. The male is the donor, the female is the receiver. So all the male needs is a sex pillars, right? The male needs, or rather the donor needs that sex pillars. That sex pillars or pili is what makes contact with the female cell. And once it makes contact through a series of processes, these cells, they're able to fuse. They share their DNA strands, and then they both live there feeling, yes, I am now resistant to nitrofurant, right? No, so that's one mechanism, conjugation. What do we need in conjugation? A sex pillars. We need a donor and a recipient. Who has the sex pillars? The donor has the sex pillars. The male has the sex pillars. Right. Now let's look at transduction. Transduction, not conjugation, especially because conjugation is one of the most common ones. How conjugation you've got. Now for transduction, we have in conjugation we had a sex pillars, right? So you can say it was the man behind the screen or behind the curtains. I don't know what term you kids use these days. So you can say in conjugation, sex pillars was the man behind the curtain. But in transduction, the man behind the curtain is this thing we call a phage vector. Phage vector, right? Now, again, let us go to the diagram. I want you first to see so that once we are mentioning these heavy terms, you're good. So a phage. When you say a phage, just know it's a bacterial phage. You know, phage, bacterial phage. So a phage, in this case, remember we said a phage is what? A virus that can infect what? Bacteria. Right? No, bacteria used to think these viruses won't catch up with them. Lo and behold. So here it is. We are having our bacterial cell here. And then we are having, now in this case, the A plus is the focus. That is the unique thing in this bacterial cell that the next cell will not have, okay? So we are having this A plus on the plasmid. No, not on the plasmid, on the bacterial chromosome here. Then we have in our bacteriophage. Remember the process of a virus infecting you. We haven't done this, but number one, it attaches, it penetrates, it enters and releases its DNA, or rather not its DNA, its nucleic material. So this infecting phage, it has bound, it has said traded. Now I've released my DNA material into the bacterial cell. Now, once it has released, we know that the bacterial cell starts, right? It transcribes the phage DNA so as to make the phage proteins. But in the process, because somewhere in the DNA we had this A plus, right? Somewhere in the DNA we had this A plus. While the bacterial cell was synthesizing the phage stuff, accidentally there was that gene, that A plus gene that accidentally, you can say, it also got replicated or transcribed, whatever you want to say, and got packaged into this phage. Now you can see all of these phages, these are normal. These ones, once they go to another cell, nothing happens, right? These are normal. But on this one, this bacteriophage with this A plus, once it goes to another cell which does not have the A plus, and it releases its DNA, it will also release this gene, this A plus gene. This A plus gene will go to the bacteria and be incorporated onto its normal gene. And now the bacteria will also have that A plus gene. So again, let's go back to our narrative. 2 E. coli. One is sensitive to nitrofurantoin, one is resistant to nitrofurantoin. So let's assume again the A plus is the resistant gene. And then unluckily, the E. coli was just walking around, unluckily the bacteriophage caught it, infected it, but the E. coli said, I am not doing what? That the world will not forget me. I have to leave a piece of myself in this world, right? And this piece, it decides to leave this gene for resistance to nitrofurant. When this is the gene it left behind, and it left it inside the bacteriophage. So now, when this bacteriophage with this gene for resistance to nitrofuran, another E. coli that initially was sensitive to nitrofuran, and it affects it, this gene is transferred from a normal cell, from a resistant cell to that one of a nini cell, a sensitive cell. Now, that's it for the generalized transduction. For the specialized transduction, essentially the same thing. Okay, the same story, just that here we are having, now here it is again. Now in this case again, the S plus gene, right? There is this UV induction, and once there's that UV induction, there is normal okay, for the specialized okay, I didn't see it being discussed in many books, but let's just look at it here together. So I didn't really focus bacteriophage and the curtains. In conjugation, the man behind the curtain was the sex pillar. It's the one that was facilitates. Okay. Now, lastly, at the very last, we have transformation. Right? How? Because transformation is literally genes are transferred from one cell to another cell, but it's just the DNA. Like, we're not in transformation, unlike in transduction and conjugation, where we saw a man behind the curtain. Here we don't have a man behind the curtains. Okay, it's just that what happens essentially is that DNA will cross the cell membrane. Like, in short, we will have just somehow we'll have a free disease that DNA is somewhere floating around, gets a recipient cell, and it enters inside that cell. And once it enters inside that cell, it's able to integrate itself into the what? Into the genetic code of this new cell. And through that, we are able to bacterial, we're able to now get the third process. No conjugation, please, no conjugation. For transformation, we even saw it when we were looking again at biotechnology. When we say transformation, literally is the introduction of a foreign DNA into another cell. Simple as that. In transformation, and essentially that's there. I just want to diagram for that, and I believe we will need. Once we see the diagram, it will be just easier to enter in another cell. It is, yeah, here is bacterial transformation. Yeah. Remember the restriction enzymes, what do they join? Right? Restriction enzyme cuts, ligase enzyme join. So here in bacterial transformation, this is from one bacterial cell to another. There was a restriction enzyme. We cut this gene to be transferred, and then there was this DNA entered, this naked DNA entered into this cell, and then DNA ligase joined that to this existing cell. Simple as that. Transformation is what happens in biotechnology, right? Once we say insulin, once E. coli, we get the gene for insulin from pancreatic beta cells, and we inject it into the E. coli cell. That is transformation. We've introduced naked DNA into this other foreign cell. Or rather, we've introduced a foreign DNA into another cell without using a sex pilus, without using a vector. That is transformation. In short, we've transferred from one to the other. I think that's it. Unless any questions, additions, by the way, if someone has additions, it really helps. I know Stanley spent, I think, half the day reading bacteriology. He may have something more. We can take a 8-minute break. At 9, we do gram staining and we start gram-positive organisms. Staph, strep, and enterococci. That one will be done by 10. We'll all be in bed by 10:20. By the, yeah.
I just want to add something.
Yeah, go ahead. Shin.
Yeah, so I don't know if we talked for the requirement for growth of bacteria. So I just want to add that when you're doing for the bacteria to grow, you either need physical or chemical, or I mean, you need both. So if it's physical, you just give me a minute. Yeah, so in physical, you'll have temperature, pH, and osmotic pressure. Now, in temperature, some microorganisms are classified into 3 groups on the basis of preferred temperatures. So some are cold loving microbes, so that will be they'll grow between 0 to 15 degrees. Those are known as psychrophiles. Then mesophiles are moderate temperature loving microbes. So they'll be between 25 to 40 degrees Celsius. We also have thermophiles, and they grow between 50 to 60 degrees Celsius. Then for pH, we know that most bacteria grow best in a narrow pH range. So it's between 6.5 to 7.5. And when the bacteria are cultured in the lab, they often produce acids that eventually interfere with their own growth. To neutralize the acids and maintain proper pH, chemical buffers can be added to it, like peptones or amino acids. Then osmotic pressure. High osmotic pressures have a effect of removing necessary water from the cell. So when a microbial cell is in a solution, when the concentration of the solute is high, the cellular water will move out of the plasma membrane. That's like hypertonic. And then it will lose water, so this causes plasmolysis. Then in for chemical requirements, you have carbon. Basically, it's just going to give energy or get energy from proteins, carbohydrates, and lipids. And they derive their carbon from carbon dioxide. Then nitrogen, sulfur, and phosphorus. It's it's mainly used for protein synthesis. Then you also have oxygen that we talked on the energy, obligative aerobes, those anaerobes and all. Yeah, that's it.
Thank you so much, Rina. Thank you for that. This is so nice. Yeah, you see when different people have read different stuff. It's in a way, when people learn on, you get to learn more.
Yeah, that's the whole point of group studies, so that you can add. Yeah, in case if there's a MCQ or anything based on it, you can just have an idea.
Guys, so for just a few minutes, we'll go through gram staining, then we'll start on staphylococcus. Yeah, so gram staining, right? No, gram staining, we know it classifies bacteria into two, right? Gram positive and gram negative, and it's the most commonly used test for identification. Why is gram staining good, or rather, even it helps so much? Because number one, you're able to see the size, you're able to see the shape. So with gram staining, remember we always say lab, or rather laboratory investigations, they are not done on their own, or rather by themselves. You pair lab investigations with clinical symptoms or clinical presentation. That means if someone is having symptoms of, for example, a UTI or an STI, or kind of having a mix of both, then you're able to take a sample and take it to the lab just through gram staining. If we see rods, right? If we see rods, we can say, hey, this is more likely to be E. coli, right? Because you paired that information with clinical presentation. If we see cocci, right, shape, we can think that this may be Neisseria gonorrhea, because gonorrhea is round-shaped gonococci, right? We'll see all this. So gram staining is very important, and always remember, just as a principle, not even just on gram staining, is that lab investigations, they are not to be done alone. You pair lab investigations with clinical presentation. If someone just came and gave you a lab presentation and they didn't tell you how they are feeling, you can't really do much. First of all, you have to know what they were presenting with, right? So in gram staining, as I've said, it can be used to differentiate bacteria between gram positive and gram negative, and it's the most commonly employed diagnostic identification. And so we know it is based, or rather, it is based on the thickness of that peptidoglycan layer. That's the key thing with gram staining. That thickness of that peptidoglycan layer. Let us look at the principle. Remember, we said the peptidoglycan layer in gram positive, it's thick, right? But in gram negative, it's thin. And then now in gram positive, in gram staining, first of all, even before going to the specific procedure, just want us to know, number one, there's a primary stain, right? Primary stain is crystal violet. And then there's a mordant. The mordant just fixes the color, or rather the dye, which is this crystal violet, right? Right? So the primary stain is also called the primary dye. So there's a primary dye, which is always crystal violet. And then the mordant, which is iodine, it fixes the dye. It fixes that, it makes sure that this crystal violet has really been, absorbed or taken up, whatever you want to say. And then there is a decolorizing agent, which is alcohol. We use alcohol or acid. Decolorizing agent. And lastly, a counter stain, right? So this is a principle in terms of, there's a primary stain. You can see once you put the crystal violet, they both were blue, right? They both were blue. We fixed the iodine, we put in the mordant, but then when we decolorize here, this is where the change comes. In gram positive, they retain the crystal violet color, but in gram negative, because that peptidoglycan layer is not thick, it hasn't taken up as much crystal violet. It's easily decolorized. That's why when you put the counter stain, there is saffron, gram negative does take up the saffron. So now let's go back to the principle, because I want us to first see the photo so that we can go to the theoretical principle. Remember, we've said number one, there's a primary stain, which is crystal violet. We have the mordant to fix the stain. Number three, we have a decolorizer, right? Which is alcohol or acid. Then number four, now we have the counter stain. So here it is that once we've put the crystal violet dye, let's go back here. I just wanted to take it slowly. Once we put the crystal violet, both the gram positive and the gram negative, they've taken up the crystal violet. I think even at this point, you can't differentiate these two. It's exactly the same color. You know, you can't say that the gram negative has taken more stain than the gram positive. No, you can't say that, right? They are all, you can say they are all the same level. They've all taken up kind of the same level of dye, right? We already saw the structure here, right? Flagella there, the pillar, in the outer membrane perisplasmic space. This is nice. Or as this decolorizing agent or decolorizer, when you have the alcohol or the acid, what does it do? It literally is, as I said, it will be harder to remove the dye from the thick peptidoglycan layer of gram positive as compared to the one of gram negative, right? Here in the gram positive, you have a thick peptidoglycan layer, so removing that is hard, right? But the gram negative is just a thin one. Remember, we said gram staining is all based on that peptidoglycan layer. That is what brings the difference when it comes to gram stain. So able to remove or the dye, or rather to decolorize it from the matte from the gram negative thin peptidoglycan. Once we add the able to see gram negative appearing, remember saffron is red. It's also called red saffron. The gram negative appears red or pink, but the gram positive, we see it still appearing a bit purple, right? Bit purple, because purple is, you can say it's this color here of crystal violet, right? You can say that color. Just know that it is based on that the thickness of the peptidoglycan layer. So yeah, so just looking at the procedure, but from the procedure number, because we don't have as much SQs, make sure you've memorized that the primary stain is the crystal violet and the secondary stain is the red saffron or the counter stain. Then the mordant is iodine, mordant iodine, decolorizer alcohol, right? So just looking at the steps quickly, we have the crystal violet, right? They both take up that crystal violet. We add alcohol, we add the mordant first to fix the stain, fix the dye, the crystal violet dye. We add the alcohol to do what? To decolorize. Then finally, we add the saffron in the counter stain, and we're able to see the difference. Yeah. So that is it for gram staining. Unless someone has something to add. Yeah, like alcohol, dehydration. Yeah. So essentially, that is it for gram stated, and the other one that is there is acid. The acid fast stated is just used to identify acid Mycobacterium tuberculosis, and that one we won't be covering so much of it, but what I want you to know just from it is that this Zil-Nielsen that is used as a Zil-Nielsen state. Otherwise, that is it for me for identification. Now let's go to took the exact 10 minutes. Yes. Now let's go to cocci. Then we can call it sesh. Call it a sesh. So staphylococci, right? Staphylococci, the most common one we know is. Then the other one we have is Staph epidermidis, and the last one we have is Staphylococcus. It causes UTI, right? But for aureus, number 2, also in food poisoning, when we're not serving hygiene, toxic shock syndrome, when we are looking at tampons not being used, or rather there have been an infection there, the tampons and all such things, right? Staph aureus, you can't run away from Staphylococcus aureus, right? So looking at its names, cocci means rats, but we will look at them when we encounter them. So for cocci, it means a round shape or a spherical shape. We know Staphylococci, number one, they are very resistant to antibiotics, right? We invented methicillin. There was methicillin resistant Staph aureus. We invented resistant Staph aureus. We invented linezolid to fight that. Now we even have linezolid resistant Staphylococcus microbial resistance, right? Judicial use of antibiotics. There's no more judicial use for antibiotics. A child develops a simple rash, the parent runs to the pharmacy and ambicloxacin. You wonder why Janet is even in this discussion once we start mentioning resistance. Is she not embarrassed? Moving on. So let's look at the general characteristics of the Staphylococcus family. Number one, I think we all know it's gram-positive. We said today we are, but two, you can say it's spherical shape, right? And then they also tend to occur in bunches, like grapes. You can see here, how they are occurring. It doesn't occur on its own. They occur in bunches, like grapes. Why, Nene? This how bacteria occurs is very important, because once we are looking at something like streptococci, we will be able to appreciate Streptococcus pneumoniae. I just want you to see a picture of how Streptococcus pneumoniae is another name for it, nini, that is said about it is that is this diplococca, because twos, you can see here, two, right? You'll have gone into two, yeah. So how the bacteria appears is important. So in Nini, we are having, you can be told in the microscope, the laboratory technician view the spherical shape appearing in bunches like grapes. What is the most likely organism? Is it Staphylococcus aureus or Streptococcus pneumoniae? Pneumonia appears double. That's what we call it, diplococci, right? Then another characteristic is that they're facultative anaerobes. Facultative anaerobes, right? Facultative anaerobes. Remember what we said about facultative anaerobes and as compared to obligate anaerobes. The other thing, they produce catalase, right? So they are catalase positive. And this is one of the features that distinguishes them from streptococci. When you're looking at streptococci, in streptococci, we won't mention anywhere that streptococci produce catalase. But staphylococci does produce catalase, right? Then the Staph aureus, specifically the Staph aureus, it does produce coagulase, or rather, you can say is coagulase positive, right? So just know that Staph aureus does produce coagulase, but for the Staph epidermidis, Staph saprophyticus, they do not produce the coagulase enzyme. Okay, so know that Staphylococcus aureus does produce this coagulase enzyme. What does coagulase do? It just causes plasma to coagulate, you know, it's there. Yeah, so now let us go to, we've covered number one, gram positive, branches, catalase positive, Staph aureus. We also seen it, coagulase positive, especially the virulent staph. Now let us cover the oh, and the other thing, we didn't mention this, but just in a gist here, in terms of normal flora. I believe when we say normal flora, we all know, you know, in our cells, in our body, I think bacteria outnumber our cells by, I think, 2 million. Right? In short, in your body, you have more bacterial cells than you have human cells. So you can say you're more bacteria. If you're more bacteria than human, then all you guys are bacteria. I'm not trying to insult you guys, but essential. Anyway, Staph aureus is a normal flora. That means it does live on the or rather, a normal flora, essentially it means it exists in our body without causing an infection, not an infection, without leading to us having symptoms. Because in microbiology, there are terms such as infection that arise. Remember, infection is as simple as being exposed to that particular microbe. So, like we know, once we say Shrina is infected with TB, it doesn't mean she is weight loss, all of these things. We just mean their Shrina has been exposed to TB. If we go to her lungs, we can find Mycobacterium tuberculosis. That is how we define infection, right? It's more of exposure, because there's these questions that also come in terms of, especially when we're looking at the likes of Kina amoeba and Amoeba histolytica, that what infects you is the cyst. right? Because that is what you ingest. That's what, but I will confirm. Yeah. So normal flora is just this bacteria that they are there on your skin, but they are not causing you to have any symptoms there. And they are all over in the mouth, on the skin especially. We have Staph aureus. On the mouth, we have some anaerobic organisms, the likes of bacteroides species. That is why when you always go for a dental procedure, after the procedure, you'll see most people are given metronidazole because of these anaerobic organisms that do exist in the mouth. You know, metronidazole, very good in terms of that. You also know in your stomach, especially in your stomach, we have some other normal flora. I think Stanley is always telling people to take yogurt. Even in the vagina, there's again normal flora, right? Likes of lactobacillus and then such things, right? So just when you have time, just go through this normal flora, just familiarize yourself with the normal flora of the different parts. So when we say normal flora, we mean that it's usually there, right? So we know Staphylococcus aureus does live on our skin. That is why once you guys are injecting people with that depot, first of all, you'll need to swab, because if you don't swab, you will have penetrated the skin, and that staphylococcus can also enter the skin. Once it does enter the body, that is when you see the true colors of Staph aureus. But when it's in the skin, it's just like, yeah, you know, we are friends. Yeah, such things. So Staph aureus, I think I've already even just alluded to this. Body, we'll see it even being implicated in things like infective endocarditis, when you're putting in valves, when you are catheters, you know, all of these things. Yeah, we remember colonization, such things. So essentially, epidemiology, we've already said, it's there on our skin, right? Normally, it's just there on our skin. But you can also get it from fomites such as adonal surfaces, such things. That's why if someone unhygienic is cooking your food and there's some Staph aureus not washing their hands, you can end up getting food poisoning and all this stuff. Then pathogenesis of Staph aureus, or rather, when we're looking at pathogenesis, mostly we are looking at the virulence factor. What causes the disease? What causes this organism to make you sick? Right? That is essentially what we mean when we say a virulence factor. Right? A virulence factor is what will make you get sick. And Staph aureus does produce toxins when we are seeing it being implicated in food poisoning. We'll see it does produce toxins and all. We can't go through all of them. Why? Because at the end of this week, we'll have seen many bacteria, and they all have virulence factors. So we can't go through each and every virulence factor. But we have a simplified diagram here that at least for Staph aureus, maybe we can look at. Yeah. For Staph aureus, we're seeing it as protein A, which is a component of the cell wall, and it binds to this moiety of IgG-Fc. We haven't looked at antibodies, but there's two portions, Fc and portion. This crystallizable portion is where that antibody will bind to something like a phagocyte, such as neutrophil, causing phagotization. But if this protein A binds to the Fc portion, then phagocytosis, whatever, doesn't take place. This fibronectin binding protein, all of these things. Yeah. enterotoxins, especially when enterotoxins, whenever we entero, we look at the GIT, we'll say the enteric nervous system, food. When you look at enterogermina, like as a name for probiotics and all of these things, just means relates to the stomach, right? Enteroingestion, it does have enterotoxins, and they are the ones implicated when your food poisoning is associated with Staph aureus, this toxic shock syndrome, toxin 1, yeah, all of this. Then when we look at the clinical significance, when you're looking at clinical significance, we're just looking where in the body can Staph aureus lead to disease. Number one, we've already mentioned, we have the skin, right? Skin cellulitis, impetigo. We know it's the most important. Once it enters your bloodstream, it can infect your bone, it can infect your valves, endocarditis there, osteomyelitis. It can also infect your urinary tract. Food poisoning, we've mentioned, it can also cause pneumonia. It hasn't been shown here, right? Staph aureus, we'll see this bacteria, when they go somewhere, they are more likely to cause infection. So it is sepsis, pneumonia. Here it does cause pneumonia, toxic shock syndrome. We'll see that, right? Is all of this scalded skin syndrome, all of that mentioned. Then let's go to identification. Number one is gram positive. You're experiencing gram positive. They are appearing in a grape, like in bunches, right? Then if you do, it is coagulase positive. Then the other stuff, the Staph epidermidis, the saprophyticus, they are negative. And that's how you distinguish it from Staph aureus. Produce this coagulase. It will make those clots when you expose it to plasma, right? Immunity, Staph aureus doesn't really elicit a strong or long-lasting immunity. And that is why at the end of the day, when we're looking at treatment of Staph aureus infection, if it's methicillin sensitive, we will see drugs such as oxacillin, clox. These days we're really dealing with methicillin resistant Staph aureus. Here we have the likes of vancomycin coming through. We have the likes of doxycycline coming here. We have cotrimoxazole, all of these. But the one specific one, they like this question aureus. It's it's vancomycin. Vancomycin is what we say is more of like the first line in terms of when we mention methicillin resistant Staph aureus. Here we can see the antibiotics I was mentioning in terms of the community acquired. With some even cotrimoxazole, you're seeing cotrimoxazole there. I don't know where doxycycline is. All of those, when it's vancomycin resistant, you want to give this person linezolid, right? Linezolid is a good one, it's a strong one, it's a last line. Moving on to coagulase negative staphylococcus. So coagulase positive staphylococcus is just Staph aureus. Coagulase negative is Staph epidermidis, Staph saprophyticus. Staph epidermidis, mostly it will just cause skin infection, but it can also infect the valves, right? It tries to live up to its brother Staph aureus. It can also infect the valve. We'll see it when we are covering infective endocarditis again. We'll see this. Saprophyticus, we already mentioned saprophyticus. It can cause what? UTI, right? Cystitis in women, right? So it's another thing. Yeah, and that is it about skin infection. We said pneumonia, we said endocarditis, food poisoning, osteomyelitis, sepsis, toxic shock syndrome. We've mentioned all of those. With staph epidermidis, skin infection, infection of the heart valves, catheters, saprophyticals, and cystitis. And you're seeing in all of these, the first lines we are seeing is the quinapenicillins. Remember, we said with gram-positive, we will note in gram-positive all around, that in gram-positive penicillins, or rather beta-lactams, beta-lactams are our number one. Beta-lactams are our number one. Also, they are facultative anaerobes. Again, just to remind you, Staph aureus, coagulase positive, catalase positive, right? Then we saw all those other characteristics. You can see the type of infection it causes here in vertigo. A boil, boil is one of the most common things you see people in the community complaining about, folliculitis, all of these things. Yeah. So, yeah, that is it. Unless someone has a question, we'll move to streptococci. Or an addition, by the way, an addition question. No question because of time, we'll just move to streptococci. Sorry guys, yeah, I got disconnected for a second there. So streptococci, number one, they are catalase negative, right? We already said that when we're looking at Staph aureus, they are catalase negative, they are gram positive, right? Yeah, they are gram positive. Again, most are facultative anaerobe. They're able to grow in the presence or absence of oxygen, right? But even when oxygen is there, they prefer fermentation, such things, right? And for streptococcus pyogenes, will we call it group A streptococci. It's just strep pneumonia, and yeah, we know strep pneumonia for any pneumococci. We've already said it can also be called pneumococci because or diplococci because it appears in so remember we just have three major ones: pyogenes, Agalaceae, and pneumococci. Number one, before even we look at the hemolytic properties, when we're talking about bacteria, we look at three things when we're looking at general hemolytic. And this relates to what will happen once an organism is exposed to red blood cells or to actually hemoglobin in itself. So with alpha hemolytic organisms or alpha hemolysis, essentially what will happen is that there'll be a partial lysis of hemolysis, or partial lysis of red blood cells, not of hemolysis, or we can say partial hemolysis. But in beta hemolytic organisms, they do complete hemolysis or complete lysis of red blood cells. But for the gamma hemolytic organisms, they do not, they have no hemolytic property on RBCs. So beta hemolytic, complete, alpha, partial, gamma, no hemolytic property. And this is how we'll be looking at streptococci when we are looking at the three. Remember, we'll be focusing on group A, group B, and strep pneumonia. Group A, group B, and strep pneumonia. So let's start with group A. Number one, group A, conceive in there, it's a beta hemolytic streptococci. Beta hemolytic meaning there is complete hemolysis, complete lysis of RBCs, right? It completely lies. I don't know if there's a diagram of a media that has undergone complete hemolysis. If someone can just Google that and send it on the group, how the different media will appear. One that has gone complete hemolysis, one that has gone partial hemolysis, and one that has undergone no hemolysis at all. Yeah. So let's start with group A, streptococci. We will also call it streptococci pyogenes. Pyogenes. When we see streptococci pyogenes, that causes the not tonsillitis, pharyngitis, right? Pharyngitis, sore throat. If it's infection, it's this Streptococcus pyogenes. And when we're looking at Streptococcus pyogenes, we'll because of its ability to cause that rheumatic heart disease, we look at one specific virulence factor that is relevant when this organism does cause rheumatic heart disease, right? Again, it does cause that rheumatic heart disease. And also for the purpose of oh wait, okay. Let's just move on. I'll look at that when we are going ahead. Okay, so streptogenes. Already by it being a streptococci, you know, sometimes general knowledge really helps. When you ask characteristics, you can know it's gram positive, right? Number one, it's ferric. Staph aureus is catalase. Staphylococcus is a catalase nini positive. You can say this one is catalase negative. You see these characteristics. So the other one now we are adding on to group A. Group A streptococci is that it's beta hemolytic. That there is complete lysis of RBCs, right? There's complete lysis of RBCs. Then the other thing is this is the organism. In movies, I know it's usually for made in movies that this is the organism that is also called the flesh-eating bacteria, right? The flesh-eating bacteria. That is this group A streptococci. That thing that causes pharyngitis, a sore throat. Is the same thing that does cause that fasciitis, necrotizing fasciitis, flesh-eating bacteria. Factor. I just want us to look at this M protein, because this M protein is what we will see it being implicated in terms of rheumatic heart disease, right? So before you see that, for clinical significance, we will say it says the pharyngitis, it does cause pharyngitis. In impetigo, erysipelas, sepsis. You can always throw sepsis for any bacteria. Necrotizing fasciitis, we've mentioned all this. This is group A streptococci. Acute rheumatic fever. So in acute rheumatic fever or in rheumatic heart disease, what you are having is, we are seeing this is an autoimmune disease that does occur 2 to 3 weeks after initiation of pharyngitis. It's when your autoimmune, meaning your immunity is attacking itself. Your antibodies will be attacking your heart valves. Why will they be attacking your heart valves? Because there is the antigens of your heart or the antigen of your valves and this M protein that we've mentioned in group A streptococci. There's that cross reaction. So the antibodies will be seeing your heart valves as if it's a foreign organ and starts attacking. So that's all. Know this disease is caused, but remember, group A streptococci, this is what causes the rheumatic heart fever or rheumatic heart disease or acute rheumatic fever, pharyngitis, necrotizing fasciitis or flesh-eating bacteria, if you want to call it that, right? Acute glomerulonephritis, for example, if there are these cases, a child was sick, right? The child just had a sore throat, and then a few weeks later, the child is having kidney failure, right? Acute glomerulonephritis, streptococcal toxic shock syndrome, all of these things. When we come to lab identification, remember beta hemolysis. I hope someone has sent a picture of beta hemolysis on the group. Yeah, antibiotics for this antibiotics. Just think of it in terms of pharyngitis. What do we give for pharyngitis? One can give penicillin, penicillin V, amoxicillin. If the patient is allergic to penicillin, azithromycin, right? The antibiotics are used. Prevention of the, why do you need this recurring pharyngitis? That will be a risk factor for this rheumatic fever, rheumatic heart disease. So then we dive into group B streptococci. Right? Group B streptococci. Group A, we've seen it, strep pyogenes, right? Group B, now we have this strep agalactiae. Now for group B, for strep agalactiae, or rather, let me just call it group B, streptococci, the most common thing it causes is meningitis in neonates. Meningitis in neonates. It is. That's its unique thing. It's a leading cause of meningitis and septicemia in neonates, right? So that's the major thing I want you to remember with strep agalactiae or group B streptococci, that it does cause meningitis in neonates. Okay? It does cause meningitis in neonates. So then moving on to strep pneumonia. So strep pneumonia, I believe when you see strep pneumonia, you can mention at least two diseases that it causes: pneumonia and also meningitis, always through otitis media there. You know, this otitis media, pneumonia, and meningitis, these are interrelated diseases. You see the same organisms that can cause otitis media, the same ones that can reach the brain and cause meningitis. Now, unlike its two big brothers, group A and group B, whereby those ones, they are beta hemolytic. For strep pneumonia, this one it does cause alpha hemolysis, meaning just partial, right? Just partial, right? And the other thing we mentioned unique about Streptococcus pneumoniae is that they occur into two diplococci. That we'll also call them diplococci. We'll also call them diplococci. Again, as we say, this in every organism, we will always see virulence factors. So we can't go through each and every virulence factor. Treatment when it comes to strep pneumonia, again, these are gram-positive organisms. You see the beta-lactams, right? Kina penicillin G. For pneumonia, we know these days ceftriaxone is given, right? Ceftriaxone, even the likes of the augmentin, the amoxicillins. We will be seeing these beta-lactams a lot. Beta-lactams are our first line of war against this the likes of gram-positive organisms. Another thing these days, because of development, there's vaccines for the elderly when someone crosses that threshold of 60, and also in kids also. The injections are given. So for ensure your children and your parents, to these vaccinations, they are good. Finally, we'll finish with enterococci as promised. Let me see. Enterococci, then Ermi said, told Sonia, we were told they recently they are questioning diphtheria a lot. So on your own time, just go through diphtheria, because it's also a gram-positive organism, but we won't get to go through it because of time. Tomorrow we'll be doing the gram-negative. So on your own time, just go through diphtheria. So for enterococci, these are same gram-positive, right? Spherical shaped, right? Then when you hear the term enterococci, that always tells you this thing is somewhere in the intestines. You can't miss it somewhere there. And same as the streptococci. For enterococci, we'll see even the term of hemolysis: alpha hemolytic, beta hemolytic. Remember, beta hemolytic, that is complete lysis. Where did we see beta hemolytic? Group A streptococci and group B. Group A streptococci was which one? Strep pyogenes. Strep pyogenes, notorious for what? Pharyngitis, rheumatic heart disease. Yeah. Why rheumatic heart disease? Just say somewhere in your explanation, so long as you mention the M protein, you'll be good. Rheumatic heart disease is an autoimmune, right? Then group B streptococci, we just mentioned, strep galaxiae, again beta hemolytic, notorious for what? Meningitis in neonates, right? And then we mentioned our friend strep pneumoniae. Pneumonia, we saw unique characteristic. Number one, diplococci occurs in two tools. It always goes in two. Other thing we saw, it's a breakdown of the RBCs, notorious for what? Pneumonia, from the term pneumonia, and also what? Meningitis, otitis media. But for this one, we saw there's a vaccine, pneumococcal vaccine. Now we're coming to enterococci. Enterococci, again, they are just brothers, these things. Enterococci there are two important ones. Just enterococci fecalis and enterococci fascia. Same, they can be alpha hemolytic, beta hemolytic, or gamma hemolytic. Remember, gamma doesn't do anything to the RBCs. So they are notorious in causing what? Nosocomial infections. But normally they do form part of the normal fecal flora. Remember, just when you see entero, you can always gamble with it being somewhere in the GIT. So we are seeing it being part of the normal fecal flora, and that we've seen they are rarely virulent. They rarely cause disease, but in some conditions, we'll see it being causing disease. We'll see enterococci can cause UTI, this enterococci fecalis, carditis, and such things. Yeah. Then there are something unique about enterococci is their ability to survive in the presence of bile. Remember, these ones, unlike strep pneumoniae, which enterococci lives in the bowels, right? That means it can survive bile, but the others cannot. Then for enterococci, something unique about them as compared to their brothers is that enterococci, they are naturally resistant to beta-lactam antibiotics and aminoglycosides, but they are sensitive to the synergistic action of a combination of these classes. What is that saying? Simply put, enterococci, if you try to hit it with a beta-lactam antibiotic alone, it to the center. If you try to hit it with an aminoglycoside alone, it will return that bullet to the center. But once these two come together, hey, enterococci will say rest in pieces, rest in pieces. It cannot withstand the combined forces of ampicillin plus gentamicin. This is a common regimen. This ampicillin plus gentamicin is there part of what is causing the infection. Know that we have to add ampicillin there. Ampicillin and gentamicin. But again, we'll see that mostly we'll see it being what we've mentioned, that it causes the sometimes just leads to a UTI and also endocarditis. Yeah, but not so virulent. That's why you don't sort of famous nini bacteria. Lastly, viridans streptococci. Here I'll just mention, I think, 3 things. One, viridans streptococci is the one implicated the most in causing what? Infective endocarditis. Viridans streptococci. Remember, there's endocarditis. Please don't confuse these two. Endocarditis and rheumatic heart disease. Rheumatic heart disease, we are seeing group A streptococci. Endocarditis, we've seen viridans streptococci, and we've seen Staph aureus, Staph epidermidis. Now, viridans streptococci is the one that is mostly implicated. And viridans streptococci, it just, number one, it lives in the mouth. That is why it is normal. It lives in the mouth. But then, I think we always say that a dental procedure risks you having a heart disease, right? Remember, in a dental procedure, how does that happen? Just in layman terms, some people come and ask, why am I taking an antibiotic after I've just had my teeth removed? Because number one, you have viridans streptococci just as part of your normal oral microbiome. Then we messed somewhere with your teeth. They may be somewhere we nicked a vein, we nicked a tiny capillary, and just that a few cells, a few organisms of viridans managed to get into your blood cell. You'll regret not having just taken that antibiotic to prevent the infection in the first. So viridans streptococci endocarditis, causing infective endocarditis. Yeah. So recap of streptococci. Number one, we say, these ones you can always throw them. Catalase negative. That one we compared it with what? Staphylococci here. Let me just take you back because we are done. We compared it with our good friend here, Staphylococci, where we mentioned that Staphylococci, we are seeing it being, and that is, yeah, is it? It is mentioned. I just want to highlight. We saw that Staphylococci here, we say they are catalase what? Catalase, what was it? Here they produce catalase, and this is the feature that distinguishes them from streptococci. Yeah, so circle shaped. And then here we brought an aspect of they have different or varying degrees. They can lyse RBCs to varying degrees, where so long as you know something, don't try to think of a characteristic, right? What you know, or use what you know. So catalase negative, yeah, we mentioned. We say they are facultative anaerobes, same as the staphylococci, facultative anaerobes. Alpha hemolytic, you can see here, this is partial, right? Partial breakdown. Beta hemolytic, we can see complete breakdown. It's broken down all the RBCs around it. Right? This. And for the beta hemolytic, we saw it's group A and group B, right? Those are the beta hemolytic. Alpha, we saw strep pneumoniae, right? Yeah. I think that is it, unless someone has any addition. Honestly, again, it's I don't know if it's leakage or anything, but Edmi has really insisted that we read the diphtheria. So in your time, go through diphtheria slowly. Yeah. Otherwise, that is it, unless someone has any additions. Tomorrow we shall be starting with our good friend Neisseria gonorrhea. I'll share the topics tomorrow or tonight that we'll be discussing tomorrow.